10x is the go-to now because you can do tens of thousands of cells all in one go. It's simplified and made it much more democratic.
I would say single-cell analysis is the default technology for characterizing biological samples.
The ability to capture a large number of cells. The data becomes much more robust when you have multiple internal replicates within each experiment. And the ability to capture several thousand cells per experiment, per run, has been a big game changer in that sense.
Essentially, single-cell analysis paired with bulk omics analysis allowed us to recharacterize, or you can say reclassify colorectal cancer. And so now we see this unified classification of colorectal tumors that mixes single-cell and bulk phenotypes, genetics, and transcriptomic into very coherent categories of colorectal cancer. And these, we believe, should ultimately be thought of as maybe even different diseases.
I think one of the things we're very interested using single-cell technology is the ability to profile in space and in time. In space, we're particularly interested in the process of metastasis. And what we've done is we've looked at primary tumors as well as different metastatic sites to ask how and why tumor cells are able to disseminate to these sites. At the same time, we are also interested in the converse, how immune cells from the distant sites, such as lymph nodes, come back and traffic into the tumor. In time, we're particularly interested in how the tumor and the tumor microenvironment changes over time with treatment, with therapeutics, with recurrences. To that, we are especially interested in the effect of immunotherapy and how immune checkpoint blockades reshapes the tumor microenvironment with regards to specific CD8 subpopulations that target tumor cells.
So spatial is really important because when you disperse the heart up or any tissue up, you lose entirely the, in the context of our heart, there is the inner surface, right, the endocardium, and then the outer surface, the epicardium. And even the myocardium, in the case of us adult humans, it's quite thick. So, you want to know whether it's on the outer surface or the inner surface. You want to know whether, and then maybe in the context of a small animal, you want to know whether it's this side of the ventricle or the other side, the septum or the free wall. So spatial, we think it's really important to reveal new insights for where these differences are lying.
If you look at the brain after drug treatment, specifically we're looking at antidepressants. What we are finding using single-cell and spatial analysis is that actually antidepressants affect the whole brain, and they affect different parts of the brain differently. And so we're studying that using model systems, and we're seeing very interesting patterns of antidepressant response.
I think, again, the ability to profile what happens over space and time really alters the way by which we think about treatment and really makes you reconsider your options in terms of physical aspects, like tumor margins, approach to this, as well as biology. What are the ideal types of treatment required?
If you're in cell biology or molecular biology, there will come a time that you feel the strong need to get into single cell or spatial, because that really is the way to help you understand and dissect out the biology.